plotting for you, this line that's rising and it's going to get to some threshold. I'm going to show you the neural basis of this idea today. And so if there is such a variable that rises at some rate, and that rate has some variance to it, so in some trial it's shallow, in some trial it's steep, well, what you're going to get then is that some trial you're going to move early, some trial you're going to move late, and that distribution is going to have a minimum out here, a maximum out there, and it's going to have a distribution that looks like this. So you're getting this skewed distribution from one possible reason for it is because you have a random variable that has a variability and it gets to the threshold. So we talked about how utility may be associated with the strength of this rise, that if you have something that is very valuable to you, you have this rise take place faster than something that is less valuable to you, and therefore you will react faster to that more valuable stimulus than a less valuable stimulus. So this was the theory that we started with. Okay, do you have any questions about this? All right, so let's now get to the brain. So here's our little cartoon of the brain. What we have is that we have this scenario where you're looking at this dot and there are these four things that appear. So here's your fixation point. There are these four stimuli, and then you're going to decide to make a saccade. Say that you decide to make a saccade to this face over here. So I want to remind you again about the events that take place in the brain when you see these things, from your retina to your visual cortex, the parietal cortex, the frontal eye field, the superior colliculus. And today we're going to focus all of our attention on this structure called the superior colliculus. I'm going to show you the activity there and how it relates to that variable that's rising. So your retina gets this image. That image is sent via a direct pathway to the thalamus, as well as the structure in the brainstem called the superior colliculus. And then from the thalamus, it goes to the visual cortex, the posterior parietal cortex, the frontal eye field, and we also have a structure in the middle of the brain called the basal ganglia, all of which is going to converge onto the superior colliculus. Now, the superior colliculus eventually is going to generate the command that says make the movement. That motor command is going to go to the motor neurons, but also is going to be sent to the cerebellum and then correct it, and then the eyes move. So the process of you deciding I'm going to make a movement from my eye from this point to this point involves a whole lot of activity in the brain, from the retina to the thalamus to all of the cortex and the basal ganglia acting on the colliculus sending its commands, eventually reaching the motor neurons, and then a copy of it going to the cerebellum and also affecting those commands. So it's an incredible amount of stuff happening in your brain when you simply move your eye from one place to another. And what I'm going to show you today is this concept of how do we look at the activity in the colliculus with respect to that variable that's rising. So we're going to talk about how there seems to be this part of our brain that is going to increase its activity, and that rate is going to have something to do with that utility, and how does it encode these ideas that we've been talking about. Eventually, that's going to become a command that's going to go out to the motor neurons, and then some other structure, in this case the cerebellum, is going to have something to say about those commands. And we'll see what that means in the next few lectures. All right. So let's remind you about the map of the colliculus. So the colliculus is a representation of the visual world because it gets direct inputs from the retina. And so it has a map of the visual space. And I want to show you what that map looks like. So here's your retina. Here's where the fovea is, and here's the space around it. This is to the right, this is to the left, this is up, this is down. And so here's, say, that the images that are appearing on your retina. You have the chair over there, you have the upside down face, and so forth. So how does it look like in the colliculus? So about 80 milliseconds or so after this stuff appears on your retina, you see activity in the colliculus. So there are two colliculi, one on the right, one on the left. And what the right colliculus cares about is in the left visual field. What the left colliculus cares about is the right visual field. So if one were to ask, well, what's happening when you're looking at this dot produces activity in the neurons that are up here. So the neurons up here, called fixation-related neurons, are active because that's what you are looking at. Your fovea is where the activity is because you're holding your eye at that location. So this visual map, the center of it is at the apex, this rostral part of the colliculus. Now, there are these lines here. What do they mean? Well, if you take a line that goes to the left, that maps on to the right colliculus like this. So things that are appearing here would activate cells along here. Things that appear along the diagonal, which is where the face, the upside down face is, would appear over there, over here. And then similarly for the vector here would appear down here. So you see how the left visual field is mapped on the right colliculus. And these numbers tell you along here the degree away from fixation. So here's 2 degree from fixation, really close. Here's 10 degree, and then so forth, 20 and 40 degrees. Okay? All right, so there would be the upside down face. Here would be the right side up face. All right, and on the other side, we have the left colliculus. It also has cells that care about what's happening at the fovea. So this blue dot would appear on both of these maps. And then the, I guess it's the bench would appear there and the noise would appear there. Okay, so, and these top layers of the colliculus, we have this map of the visual space. And the right visual space is on the left colliculus and the left visual space is on the right colliculus. Okay, so the superior colliculus is getting direct feedback from the retina. And on animals like frogs, that's how they move. The bright light goes on, they hear a sound, and they make a movement. Yeah?
Does the visual field closer to the fovea get, is it
yes.
neurons dedicated to it?
Yes, yes. You see it in this here. From 2 to 10, it's big. 10 to 20, it's small. 20 to 40 is even smaller.
So if we move our fovea to the thing that we want to fixate on, then we're going to have more information
yes.
in our visual field.
Yes.
So.
That's right.
Okay.
That's right. So the point that I was making was that your superior colliculus gets information directly from the retina. But if all you had was the colliculus and the retina, well then whatever stimulus that happened to be strong, you know, light, sound, whatever, there's also an auditory map here, would direct your attention to it. So why is it that you don't make movements to things that are, you know, just bright? Because your cortex is controlling the colliculus. So on the superficial layer, you're going to get activity that looks like the retina. But then to make a movement, that activity has to get to the deeper layers of the colliculus. And there's a whole lot of inhibition going on saying, don't move. I want you to pay attention to this thing. Keep your eye there. So the concept of utility, which is deciding among all the things that are in the visual field what's more interesting, is going to be computed elsewhere, imposed on the colliculus. So the basal ganglia is inhibiting the colliculi. It's saying, you know, just because there's all this visual stimuli there doesn't mean that you should make movements there. Something has to allow you to move, remove that inhibition. Okay, so in the intermediate layers of the colliculus, you must get activity. But those layers are getting inhibition from the basal ganglia. So at 100 milliseconds, after you've seen this, the basal ganglia is still inhibiting the colliculus. You can't move there. You can't quite move. All right, so then for you to make a movement, what has to happen is that, you know, these structures have to remove their inhibition. And at 200 milliseconds, that happens, this inhibition is removed, activity comes from elsewhere in the cortex, and now you get this activity in this location, and that location happens to correspond to this face, 10 degrees down over here. And so when there's activity here, that generates a movement. So in the superficial layer, you just have a visual space representation. In the deep layers, it's also a visual space, but it's saying, where am I going to make a movement to? So you get this action associated with activity there. And when that activity takes place, the eyes make a saccade to that location. The fovea moves. So that's the anatomy of the colliculus. Now I want to show you what happens as the neurons receive information. So overall, what we have is a scenario that looks like this. From your retina, you get information right to the superficial layers of the colliculus. But for you to make a movement, activity has to take place here in the deeper layers. But that activity is not going to happen because all of these regions of the rest of our brain is controlling what activity is getting there. That's the place where this utility is being computed. Once a particular location is activated, that then generates the motor commands. It goes to the motor neurons, and a copy of it goes to the cerebellum that controls that movement as it is being sent. So today our focus is this structure here. How do we go from the superficial layers to activity in some location on the intermediate layers? So let me show you what we know. Okay, in this experiment, what the monkey is doing is looking at this dot here. And what's going to happen is that the dot is going to go away, the fixation is going to go away, a dot is going to appear in location A. And what's happening is that David Sparks is recording from that superficial area, that place in the superior colliculus that things look like the retina, if you will. And the receptive field of this cell is here. So when the animal is looking here, well, not much is happening. As soon as this stimulus A is flashed, so here's the duration where that stimulus is on the screen, about 80 milliseconds after that appears on the screen, you see activity in this cell. Now, in the top figure, we also have eye position here. We have horizontal and vertical position of the eye. And so what happens is that about 180 milliseconds after the stimulus appears, the animal makes a saccade. So there's a saccade that he makes to the location A. In this condition, the same thing is happening. The stimulus at fixation goes away, A appears, the stimulus at fixation reappears, but in this case the animal doesn't make a saccade. So he's looking at this point. This dot appears, this goes away, this reappears. Sometimes the animal makes a saccade to it, sometimes it doesn't. But the point is, if you look at what this cell is doing, this cell behaves the same under these two conditions, despite the fact that the animal in one case made a movement, in another case didn't make a movement. So it's just saying there was something that appeared on the screen, and it was in my receptive field. It's just a visual representation of the world. It's not saying make a movement there. Okay? So it's just a visual representation. All right. On the other hand, in the deeper layers, there are neurons that are going to indicate whether the animal is going to make a saccade or not. So here's a deeper layer neuron. Its receptive field is also here. So when A appears, about 80 milliseconds, 70 milliseconds later, you see activity in this cell. But then it shuts down, and then in this condition, when the animal does make a saccade, it builds up and gives you a burst. In this condition, where the animal doesn't make a saccade, it has this weak activity, but there's no burst. So this activity is occurring at a location where if you had activity there and it reached high enough, you would make a movement to that location. But in this condition, what happened was that you saw activity there. This came because of the stimulus, but there was no build-up here to some level to make a movement. Whereas here, there was activity because there was a stimulus, but then there was build-up, and then it reached some threshold and the animal made a movement. That's the idea. Something is inhibiting the response to that visual stimulus. And then it's allowing buildup of activity to reach something, and then when it does, the animal makes a saccade. Okay, just to show you that it really looks like these cells are responsible or highly correlated to making a movement. So here's a condition where the animal makes a saccade at 100 milliseconds. Here's a condition when the animal makes a saccade at 200 milliseconds. And you see this neuron, its activity. When the animal made the saccade at 100 milliseconds, the neuron was active early. When it made it late, it was active late. In all cases, you see, so here's this buildup of activity until it reaches some firing level. And so the latency of the saccade was really closely related to when this burst occurred. You see the burst is occurring here. Okay, so the burst in this visuomotor neuron, this neuron that is sitting in the intermediate layers, always coincided with whether the animal made a saccade there. It has a receptive field, just like the visual neuron, but now it tells you whether we're going to be making a movement there. All right, so now I want to show you this buildup. That there is this buildup of activity, and when the buildup of activity reaches a high level, you make a movement. If you have the buildup of activity but it doesn't reach this high level, the animal doesn't make the saccade. So analogous to our idea that there's this rate that's rising and it's got to reach a threshold before a movement is being made. So here's a condition where you have a fixation point, a target that appears in the receptive field of the cell, and another target that appears outside the receptive field of the cell. So the simple condition where you get fixation, target, back to fixation. Boom, boom, boom. Sometimes the animal makes a saccade. Here's a condition where the animal made the saccade. Here's a condition where the animal doesn't make the saccade. So boom, 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 the animal just stays there, doesn't make the saccade. Whereas in this condition where the target goes on and stays on, the animal does make a saccade. So here's the activity in the cell that has this receptive field here. You see, when the target appears in this receptive field, it gives you this visual response, it shuts down, then it gives you this burst, and in this case, the burst isn't very strong. There is no saccade. In this condition, it also responds to the onset of the visual stimulus, then it shuts off, then it builds again, and now it gets to a high level of activity, and there you get a saccade. Yeah?
Why does it shut off if the A stimulus isn't removed?
Right. Because I think the basal ganglia is shutting off the response. So there is some initial response to the fact that something happened, but the rest of the brain must be making it so that I'm not going to respond to this until I have had time to evaluate what does it mean. Do I want to direct my attention to it? It's one way to think about it. All right. So here's another condition. Thank you for the question. So in this condition, we have the A and B targets. So we have the fixation at O. A appears, it goes away, then B appears. So A is in the receptive field of the cell. What the animal does in this case is that it does not make a saccade to A. Rather, it makes a saccade to B. So you see what happens here. So A appears. A is in the receptive field. The cell fires. It shuts down. A goes away. B appears. And then there's some activity here. And then there's some buildup of activity, but it's not very strong. So it's as if the animal is thinking about making a saccade to A, but eventually doesn't make it. Here's the condition where the A and B once again appear. So fixation, A appears briefly, then B appears. A is in the receptive field. The animal makes a saccade to A. So here's the response, and then you have this gradual buildup of activity until you get a burst. And now the animal makes a saccade to A. Even though B is on the screen, the animal makes a saccade to the location where A was located. Okay? So you see again, buildup of activity until you get a burst, and then you get a saccade. So if we were to summarize this, what we see is that in a trial where there is no saccade to A, you see this buildup, but it doesn't reach very high. In a trial where there is a saccade to A, there's a buildup and it's strong. And so then the animal makes a saccade. Yeah?
Is it twice as long in the OAD trial because it then is processing the third stimulus, so to say, B?
Twice as long.
To make the saccade?
Yeah, why does it take so long to make the saccade, you mean? So it's just one trial. And in this condition, there was these two stimuli appeared, and for whatever reason, the animal decided to make the saccade to the first one, even though it wasn't there anymore. Yeah?
Can you explain to me what the bump in both the H and V on these graphics are?
H is the horizontal position of the eye, V is the vertical position.
Okay, so there's not really any specific marker as to how far it moved. Did it just have to move a little bit or something? Because in the far right one, H barely changes at all, but it seems like you need both of them to change in order for a saccade to happen.
Well, let's see. I think this is I don't know whether this is exact. So let's see, V is down here. I guess this is just a cartoon to show you the relative, whether the relative positions are actually the case may not be true. I think I drew it as a way to show you the notion of where they are, but the actual location may be different than in fact it is different than the horizontal and vertical position. But excellent point. Thanks for bringing that up. But H and V are horizontal and vertical eye position. The timing tells you when the saccade took place. Okay. All right, to summarize. In the colliculus, cells respond to visual stimuli after 80 millisecond delay, but it takes another 80 milliseconds or more to start a movement. So this is kind of the puzzle. So if the stimulus is already there, why do you need more time? Why couldn't you just make a movement when the stimulus is there on your fovea and now has reached the colliculus? The idea is it takes time because the brain has to decide, do I want to respond to this stimulus? Is this something that I care about? And so cells in the intermediate layers have a receptive field for the visual stimulus. If the stimulus appeared in a receptive field, they begin to fire. If the firing rate builds up to a large enough quantity, the movement takes place to that stimulus. If the firing rate doesn't build up, no movement is made to that stimulus. Okay. Any questions so far? All right. So it turns out that a decision to move coincides with two events. One is the place that encodes the movement has to increase its activity, but the fact that you're not moving means that there's some activity in some place, and that's the fixation area. So when you're looking at something, you are actively doing something. To move, you have to give up what you're doing and then move to that location. So you transition between a hold state and a move state. So for you to move, you have to say the utility of this act is higher than what I'm doing now. Right? So I'm going to show you that comparison, we can understand it neurally on the colliculus. So on the colliculus, you have activity. When you're looking at something, there is strong activity in that fixation-related area. When you decide to make your move to some location, that's because the activity that's being promised there, the utility of there, is more interesting than what you're looking at. And that's what's going to happen is that the activity falls in the fixation area, the activity rises in the goal area, and then you make a movement. So two things have to happen. Not just something rises to reach threshold, something has to fall for you to make a movement. Okay? So let's see what the data looks like. So here's our colliculi. Here's the right colliculus, here's the left colliculus. So remember, up here we have activity that represents the fact that I am doing something. I am holding my eye looking at this dot. I am holding my eye looking at this face. For me to look at another face, I have to let go of this face and believe that over there, that other face has a greater utility than the one that I'm looking at. So you have to make a comparison. And the act of holding is not doing nothing. It is maintaining your focus on something. And then let's see what that looks like on the colliculus. So here's the fixation point. Here's the target. So the fixation point is on, and then the target comes on. So what happens to this activity here is illustrated in these plots. So when the animal is looking at something, the neurons up here, which are encoding the space right around the fovea, they're active. They're active, they're active, they're active. As soon as the animal decides, I want to move, they shut down. And then when the animal gets there, they reactivate. Okay? So the fixation-related neurons on the colliculus are the things that are active when the animal is looking at something. And what we're going to talk about now is what do these cells do during the time when the animal is deciding, I want to go look at the stimulus over to the right. So in general, these cells are active when the animal is holding fixation, as you see in these plots. So the animal is going to make a saccade here. Ipsiversive means to the same direction as the colliculus. So if this is the left colliculus, this is a leftward saccade. This is a leftward small saccade. This is a leftward large saccade, leftward small saccade. Contraversive means opposite. So in this condition, if this is the left colliculus, this would be a rightward small saccade, rightward large saccade. But the point is, in all conditions here, what you see is that the cell is active when the eyes are holding still, the cell shuts down when the eyes are moving, and then it goes back up as soon as the movement ends. Okay? So it's like the utility of the action. So if it's high, I'm not going to move. I got to shut down before you're allowed to change position, to move to some other location. Okay, so two events occur during what we call reaction time. That 200 milliseconds of time before you decide, okay, I'm going to move my eye over there, what's happening is that the activity falls in the fixation-related neurons and the activity rises in the build-up and burst neurons. So here's our scenario. You're looking at F, the fixation. The target can appear to the right or to the left, and you don't know where, but you know that it always appears either to the right or to the left. So you're waiting for one appear over here or over there. So it's going to appear either to one side or the other. So let's see what happens. So suppose that the target is going to appear to the same side as the colliculus. So say if this is the left colliculus, then the target appears on the left. So here's when the fixation point goes off, there's this delay period, and then the target goes on. So during this time, you're anticipating it's either going to go on here or there, right? Because that's the pattern of the experiment. I'm looking at the dot. The stimulus is either going to go to the left or to the right. So when the fixation point goes away, when this goes away, what happens is that the fixation area neurons that begin to decline their activity. You see it on both sides. Then these build-up neurons, the ones that are encoding the fact that the stimulus can be to the left or right, they begin to anticipate. I think the stimulus is going to appear in my receptive field. I think it's going to appear in my receptive field. When it actually does appear, boom, one side wins. And it directs the eyes to that direction. So the anticipation, because you know the stimulus is either going to appear here or there, is shown here in the buildup, is shown here in the fall. Both of these are saying something important is going to happen. The fixation is going to go away. The cell on the right colliculus is saying, I think a stimulus is going to appear on my receptive field. The cell in the left colliculus is saying, I think it's going to appear in my receptive field. They both are showing this buildup. When it does appear, one of them wins. And it gives you a burst. And so then you may move your eye to that direction. So those were the build-up cells, these are the fixation cells, and then these are the burst neurons. The burst neurons, they look like motor neurons. They only become active when you move. Yeah?
So the cell expects that because like it has some kind of memory?
Yeah.
How does that work?
So the rest of the cortex has all kinds of information, right? It knows where things are going to go because of all the past trials it has learned. So it can predict, here's where things are going to be, in this location. And so it reduces its inhibition for that location. It increases its excitation for that location. And that's what results in this buildup. You can anticipate when something's going to happen and where it's going to happen and when it's going to happen.
So if it sees this process that it expects to happen, then a process in the brain starts?
Yeah, that's right. That's right. So that's what your cortex is doing. The colliculus is listening to all the stuff and it's reflecting that computation. Yeah?
Lateral and contralateral target neurons are active at the same time?
Yes. Yes, they build up at the same time.
And they're both on the same side of the...
Opposite sides of the colliculus.
So if the target's on the left, then the left side is the lateral target should probably be on... Actually, no, it should be on the right, right?
Right. So, but in this condition, you don't know which one, right? That's why both sides anticipate it.
Wait, I thought the fixation... No, that's not anticipation. That's the existing...
Correct. But it knows that the fixation... Well, fixation goes away, right? Fixation goes away here, and so these begin to fall.
And each of those is on a different side of the...
Right. Yeah.
So if in the experiment you were told that it would only appear to the right?
Then only the left colliculus would go up. And in fact, in scenarios when you know it's going to appear, the activity might even reach a threshold before it comes on. So in those scenarios, you can make saccades at half the reaction time of normal conditions where you don't know where it's going to go. We'll come to that in just a second. Okay, so here's a map of the colliculus. So this is the left superior colliculus. This is the superficial layers, the ones that look like the retina. They just care about the visual information. Here in the middle, things have just a bursting pattern, and then down at the bottom is the build-up neurons, the ones that have this anticipatory activity, and then they reach a threshold to tell you, make the movement. All right. So let me show you in a little finer detail the activity in that fixation-related area. So that fixation-related area is active when your eye is holding still looking at something. It begins to fall in anticipation of the movement. So here's an example. Here's the fixation point. It's on. It goes away, and then a target is going to appear at some time. So you see that the fixation-related neuron, at about 100 milliseconds after the fixation point goes away, it begins falling. And then it keeps on falling until the saccade is made where it's almost shut off. And then when the saccade ends, it immediately goes back up. Okay, so in some conditions, the target comes on much later. And so what happens here is that the fixation neuron, it kind of the brain is expecting the target to appear here, but it didn't. So it goes down and says, well, maybe it's not going to come. So it's going to go back up, and then now it goes away, then it goes back down. So usually it would have come here. Today, it came much later, and so the animal thought, well, maybe it's going to come, fixation is going to come back on. But then, in fact, the target did appear, and so the event went down, the activity went down. So if one tracks this as a function of reaction time, what one sees is that as you increase this gap period between the fixation point and the target, you see that the fixation-related activity falls, as does the reaction time. So the animal reacts faster if this activity has fallen further. So it's like the hold signal. The hold signal is falling, and if the stimulus appears, you have a faster reaction time to it. Yeah?
These neurons are general then, they're not area specific?
Fixation, only toward fixation.
Okay,
Active.
fixation
Yeah.
neuron is related to one point in space?
Fovea.
Wait, what?
Stimulus on your fovea. So keep your eye still. What that means is keep the image on your fovea. They're active when there's something of interest on your fovea.
Okay, but it's general, it could be in any position.
Right. Because it's all depends on the retina. Where, what's on the retina. Everything is with respect to the center of it, which is the fovea. All right. So here's our summary. Before you make a movement, so here's when the fixation point disappears. What happens is that the activity in the hold area, fixation-related neurons, falls. The activity in the build-up area rises. So these are two things that goes on simultaneously, which then, at some point, the one goes to zero, the other one reaches a threshold, and then a movement takes place. All right. So we know that these fixation-related areas are important because if one were to stimulate them, so here's the stimulation here. What you see is that the animal can't move its eyes. So here's the fixation point. It goes away. Target appears. Normally, the animal makes a saccade at 200 milliseconds. But if you stimulate bilaterally the fixation area of the colliculus, you don't allow that activity to fall, the animal can't make a saccade. Only when you remove the stimulation, then the animal makes a saccade. Okay? So I want to give you a sense of these two competing things, hold and move. The hold activity is about what you're doing now. The move activity is about taking you away from that. Do something else. And these are two different utilities, and they compete with each other. And when one is becoming greater than the other, then you change. So maintain your state, change your state. Why should I change my state? Because I think there's something better there. But you see it in something as simple as the colliculus and a simple action like a saccadic eye movement. I just love this stuff. I really think It is the foundation of behavior. By understanding how these simple movements are encoded in the brain, I think we get a sense of why is it the way we are? Why is it so hard to not lose focus and not keep doing the same thing and just give up on it and go do something else? Well, because it's hard to keep maintaining focus. Because after a while, utility falls and you say, I'm not going to get anywhere. I think there's something better over the fence. I should just walk over there. And so when you do that, some utility is winning. Then it's hard to stay. All right, summarize. In the superior colliculus, cells respond to visual stimuli onset at 80 milliseconds, but it takes another 80 milliseconds or more to start a movement. If you were a frog, you would just make a movement as soon as something came, because that something was, a bright light. Maybe there's something interesting there. But you don't do that. You wait, and it takes you time to compute, should I make this movement or not? And that should I make a movement is a computation that your brain is performing, associating, well, I think there's something good over there, but I think also there's something good here. At some point you decide, okay, I think what's over there is better than what I have here. I'm going to make a movement. It takes time to compute that utility. Cells in the intermediate layers have receptive fields for the visual stimulus. If the stimulus appeared in the receptive field, they begin to fire. The rate builds up, and if it reaches large enough quantity, the movement takes place to that stimulus. If the firing rate doesn't build up, no movement is made to that stimulus. But there's also these cells, which we call fixation-related areas, which are active when you're holding still. It tells you there's something good that you're doing now. And this activity has to fall, while activity in the cells that encode the target location rise, so they're going to be inhibiting each other. And then you make a movement when these two events take place. All right. Okay, so let's get closer to this concept of latency and the rate at which these things are falling and rising. Let me give you another example of this process. So here what we have is that what's called a gap task. So there's a fixation point, it goes away, there's nothing on the screen, and a target appears, and then you move your eye. Then there's this overlap task where the fixation point is on the screen, the target appears, and then the fixation doesn't go away, and then you make a saccade. So I want you to think about what's happening here. You're looking at something, it goes away, something else appears. That's one condition. In another condition, you're looking at something, it stays there, something else appears. So which one do you think you will react faster to? Raise your hand. Yeah? Why?
Because the thing that you're focusing on initially is still there. It takes a while for you to determine if the second one is more interesting than the
Right. So if the activity that you're focusing on goes away, you're ready to do something new, right? Whereas if the thing that you're focusing on is still there and something else appears, well, I have to compare these two. And so therefore, you're going to make a faster movement earlier. You're going to make the movement earlier in the top condition, called the gap task, than the overlap task. And that's shown in the reaction time distributions here. So here's a couple of subjects, and you see that the reaction times they move earlier in the gap task than in the overlap task. Okay, so these are monkeys, but it's also true for humans. All right, so what's happening in the brain? Well, again, this is the superior colliculus. So what kind of cell is this? Fixation neuron, right? Because it's active when you're holding. In the overlap task down here, the fixation stays on the screen. In the gap task, it goes away. So you see, in the gap task, the activity begins to fall much earlier than in the overlap task, right? That gives you a reason to, well, I think you're going to make a reaction time faster in the gap task. In the intermediate layers, where the activity is encoding the movement, you see, in the gap task, it begins to rise a little bit earlier than in the overlap task. And it reaches its peak earlier than the overlap task. Big difference in the fixation-related neurons. Okay, so reaction times reflect it. Okay, so we see an example of that. Now, let's look at a decision-making task where the animal has to decide something about the location of the stimulus. And in this case, it's a really simple task. So what's going on is that there is this array of lights. The animal's looking at this fixation point here, and then it's going to have to decide when a light comes on, is it distance to the fixation point short? In that case, I'm going to make a saccade here. Or if it appeared up here, the distance from the fixation point is long, I'm going to make a saccade over there. So there's an array of lights. One of them comes on. The animal decides, is the location of the stimulus close to my fixation? In this case, I'm going to make a saccade to the left, to short. If it's far from my fixation point, I'm going to make a saccade to the right because distance was long. So why is this a decision-making task? Well, you can think about, while this is an easy decision, this is a little bit harder, this is really hard. Right? When it's right down the middle, is that short or long? What am I supposed to do? So I'm just making a decision, right? Okay, I'll move long. So again, it's easy, a little bit less easy, a little bit less easy, really hard, easy, easy, really easy. Right? Okay, so what happens? In the colliculus, so this cell has a receptive field to the right, for a saccade to the right. So where is it located? In the left colliculus. So its receptive field is for a saccade to the right. And what it cares about is when the stimulus has a long distance to the fixation point. So when the task is easy, when the stimulus appears up here, this cell is going to have a high spike rate because it was a really easy task for it to decide. So you see the activity begins to rise really early, and those are the trials where the animal has a fast reaction time. On the other hand, when it's right down the middle, then it's really hard, and in this case the animal made a saccade to the right, and you see that the activity rose, but rose more slowly. So the rate is telling us something about how hard it was for the animal to figure out whether to move to one side or the other. If it was an easy, rate went up fast. If it was hard, it went up slow. And similar for when the saccade is made to the other side. So here, the cell has a receptive field here, right? It likes to make a saccade there, but you have to make a decision. Where am I going to go? Well, at the beginning, this cell says, I think it's long. In all of these conditions, it wants to make a saccade to the right, but in fact makes a saccade to the left. It's because the other side won that race, right? And so its activity never reaches threshold. Questions? Yeah.
Are these bursts of built-up neurons visually responsive or just motor control?
They are visually responsive, but they get shut down right after the stimulus is presented. So you see a visual response and then gradual build-up. The visual response is never big enough to give you a movement. Unless it was anticipating it, then it was kind of building up already, and then a visual response comes and it takes it to threshold.
Okay.
So if in the first couple of slides, you'll see it gives a burst about 80 milliseconds after the stimulus is shown, then it shuts off. Yeah?
So you're saying that the monkey was trained to look in a certain
Louder.
So the monkey was trained to look in a certain direction?
Monkey was trained to make a decision. So the task was, a light appears. Monkey's got to decide, is that light at a short distance to fixation or is it a long distance to fixation? If it's long, then I'm going to make a saccade to the right. If it's short, then I'm going to make a saccade to the left. Does that make sense?
But it was trained to.
You trained, say again?
Because it's I'm not quite sure how it distance from the fixation point.
So say it's you. Say it's you. You're sitting in a room. Okay? I tell you, keep looking at this dot. Now, while you're looking at this dot, so here's fixation, right? The fixation is on. One of these lights is going to go on. You have to make a decision. Was that light close, in which case you're going to make a saccade here? Was that light far, which you're going to make a saccade there? Among the places that you see. So there's only 6 places or 7 places where the light can appear. And if you get it right, you get $1. So the animal learns when it's here, it's close, makes a saccade to the left. When it's here, it's far, it makes a saccade to the right. And when it's in the middle, it's only right half the time, no matter what it decides. Did that answer your question? Okay. All right, yeah.
In order to do this, don't they have to make a saccade first to see where the dot is?
No, they don't, right? Just because you are looking at my face, it doesn't mean you can't see anything out here, right? You can.
So they're able to interpret visual information that they're not focused on?
Absolutely. Yeah, you have all of this. Otherwise, how do you know when to make a saccade someplace, right?
That's tunnel vision.
Yeah. Yes, sir.
Are babies born with,
Louder.
are like very like born into 2-day-old babies?
Uh-huh.
Can they do this?
I have no idea. Sorry.
Not this child, the one before it, where it was just going left and right. Like, is it a learned behavior, or is it something you just automatically?
Do you mean orienting? Because we orient to stimuli, and the map from the retina to the colliculus to the motor neurons is certainly formed by the time we're born. So I would guess that yes, you can make, in fact you have to be able to, right? Because for the child to find the mother's breast, it has to be able to know all the stimuli associated with it. So orient, which is an orienting process, right? So this must be, yes, yeah. Probably so, yeah. And probably at birth, things like the sound of the mother, the smell of the mother, the feel of the mother, those are probably the incredibly salient things for the child, and it allows it to orient itself toward it. I had never thought of that, but good point. Okay, reward. So, only recently, neurophysiologists began looking at the effect of reward on this activity that I've been telling you. So reward alters the utility of the action. And so one can have a scenario where fixation light is on, target appears, but you can't make a saccade to it. So the target goes on and off. You have to looking at the fixation. You got to remember the target was here. The animal makes a saccade to it. Here's the saccade. Now what has to happen is that on some trials, the animal knows, I have to make the saccade, they're not going to give me any juice for it, and then hopefully in this next trial, when I have to make a saccade, they'll give me juice. But they know that on this block of trials, this saccade is completely worthless, but it's got to be done. Otherwise, they'll never move on to another trial that I might get juice for. So, some trials, the animal knows he's not going to get reward, no matter what. But he's got to have to do it. On some trials, the animal knows, I'm going to get juice if I do it. So, in both cases, the animal makes a saccade. We know he makes a more vigorous saccade in the condition where he gets juice. He likes those trials. In trials where he doesn't get juice, he makes it slowly. But nevertheless, he does make it. So, what one sees is that it anticipates it. So, it's okay, so the target's going to appear there, and if it knows that that target is really valuable, there's this buildup of activity before it appears. But if it knows that target is worthless, I wish it doesn't appear. So here it is, the activity is low. Okay, so the buildup of activity is related to the value of that stimulus. It's like anticipating something good. And if it does come, I'm going to be not only ready for it, I'm going to move earlier because I have all this buildup activity in this cell. Whereas, it's going to come, I hate it, I have to make a movement, there it is. So the activity that comes after the stimulus is lower, and the buildup of activity is higher in the case of reward. So in either of these conditions, the animal doesn't make a saccade. So some cells don't care about this reward in the buildup period. Some cells do care about it. Both of the cells fire more when the target appears if it's going to be rewarding than otherwise. So in neither of these conditions, the animal's making a saccade. So saccade is going to appear later, right? So this is just what happens in response to the target. And you see that there's greater activity in response to the target, and there's greater buildup. And so it turns out that the cells that behave like this, that care about reward, they are not in the superficial layer. So these cells are, like your retinal cells. They don't really give a hoot about the value of the stimulus. They're more down in the deeper layers where you see cells that are modulated by the value of the stimulus. That they have that buildup of activity and they fire more when the stimulus that I'm about to get is rewarding. Okay, summarize. In the superior colliculus, cells respond to visual stimuli at 80 milliseconds, but it takes longer than that to start a movement. During the reaction time, two events occur. Activity in the fixation-related neurons decline, while activity in the movement-related neurons increase. Utility of the action during the holding appears to influence the rate of decline in the fixation-related neurons. So, you know, if I'm really, this is a valuable thing, if I'm certain about it, the thing falls faster and it rises as well faster. Utility of the action that evokes the saccade appears to influence the rate of increase in the movement-related neurons. All right, so I'm out of time, and I think that's a good place to stop. I'm happy to answer any questions for you afterwards. Thank you so much.